So good morning. Thank you for coming to our last day talking about data visualization. Our keynote speaker this morning is Nils. He's an assistant professor in the Department of Biomedical Informatics at Harvard Medical School, and his research focuses on developing computational tools for data-driven biomedical research, with special emphasis on visualization. And importantly, he's also been a contributor to the Points of View articles on data visualization and nature methods that we've sort of been using as the inspiration for a lot of the content we've discussed today. Thank you very much, Nils, for talking this morning.
Thank you very much for the introduction. Thank you very much for the invitation to speak here today. Before I get started to talk about what I'm planning to talk about today, which is data visualization and discovery, and you can remove some commas or insert some colons and things like that with this talk. Really, I want to briefly say a few things about what I'm doing in general in my research. And here's just a little overview of the questions that my lab is covering. So I have a lab in the Department of Biomedical Informatics at Harvard Medical School. And so I'm working on biomedical data. But as you know, that space is pretty large. My focus is on genomic data, but I'm also working on, for example, sensor data from devices like Fitbits, or if you're familiar with the software that Apple built into iPhones and provides for researchers, which is called ResearchKit. You can collect all sorts of data about individuals using those devices. So we're working on that as well. But in the big picture, what I'm really interested in is this interface between human investigators and analysts and data. Well, the interface between the human and the machine, which is really, in our case, data. Data here, as I said, can be pretty broad across many different areas of biomedicine. And what we're trying to do is to create these interfaces between humans and machines as efficiently as possible. And one of the most efficient ways to create an interface between human and a machine when it's about data is to use data visualization, because it's a very high throughput channel, as you've probably learned. And you can ask all sorts of questions, and you can bring in the knowledge of the human investigator and analysts for data interpretation, and then come up with hypotheses and hopefully discoveries at the end. And so we've created a number of tools sort of on the human end. Of course, everything runs in a computer. For example, we've worked on visualization tools for cancer subtype discovery. That's one thing I'll cover in the talk today. We have created tools for exploration of intersections of sets. You're probably all familiar with Venn diagrams. That's something we've improved on. There's an approach called upset, which scales much better than Venn diagrams, for example. I don't know, maybe you've covered this. You did? Great. We're working on tools to visualize complex genomic rearrangements, as well as tools to visualize 3D genome interactions. So unfortunately, I don't have a screenshot of that. Now, that's the human side, and that's the stuff that's really exciting. Unfortunately, to make that happen, we also have to do a lot of work on the machine side to really create infrastructure to build visualization tools, because as you know, genomic data sets are pretty complex, large. So we're dealing generally with gigabytes or terabytes of data. Often there's a lot of complexity and heterogeneity in data, simply because we want to look not only at one genomic data type, such as sequencing data, but also at proteomics data, metabolomics data. We want to look at all sorts of different results derived from that data. So it gets pretty complex pretty quickly. And to handle that kind of data and to make it accessible to visualization tools, we created a platform called Refinery. I'll briefly touch on that in my talk as well. And one of the big things I think that you as researchers are probably familiar with is the issue of reproducibility. And that's something that sort of is part of my work, or well, it's a big part these days, because really it affects everything in here. And what that is about, I'll discuss in the next hour or so. And I really want to invite you to interrupt me whenever you have a question. We have plenty of time today. And feel free to ask whatever you don't understand, or if you have any great ideas, let me know as well. Okay. So about a year ago, Nature asked a lot of people, 1,500 researchers, if there's a reproducibility crisis in science. So lowercase s here, not capital S. That would be a little bit rude, I think, for Nature to ask if there's a reproducibility crisis in the journal Science. So they asked this question, and they got a pretty interesting answer. Has anyone seen this story? It came out around last May, I believe. Okay, so I'll tell you a little bit about it. So the respondents here on the x-axis, 3% said there is no reproducibility crisis. 7% were not sure whether there's a reproducibility crisis. 38% thought, well, there is a slight crisis. I'm not sure quite how to define slight crisis versus a significant crisis. But I'd say if we add this up, we have about 90% of all people that they asked say there is a reproducibility crisis, either a slight one or a significant one. So that is pretty striking. And they also asked, well, what are the problems? What do you see as issues with reproducibility? And here's an overview of the factors that people reported that are contributing to irreproducible research. At the very top here, with almost 100%, something that either always or sometimes contributes to irreproducible research is selective reporting. And there's sort of more social things such as pressure to publish, low statistical power. There was not enough replication, or not enough oversight and mentoring. That's basically blaming, I guess, the mentors, or a way for the students to blame their mentors. But the methods or code were unavailable. There was poor experimental design, et cetera, et cetera. Fraud, actually pretty high up on the list. It's almost 70%. People think that fraud contributes to irreproducible research. But let's look at two components in here, two of these factors. So selective reporting and code or methods not being available. Well, why is that? It could be intentional. So that's more, well, it's not fraud, but often enough people feel like not including everything they have or everything they know or everything they've done simply to make it harder for other people to reproduce their work. It's a fact. But there are other issues. For example, the inability to capture everything you've done. Right? So if you try to write down somehow everything you've done in your analysis, that actually gets pretty tedious. And even if you think you've written everything down, there are often still things that you realize later on when you try to reproduce your own work that you forgot to write down or simply didn't expect to be relevant. And finally, even if you could capture everything, do you have a means to communicate all of this? I mean, of course, you could just dump everything, like copy your lab notebook or wherever else you keep your notes about what you're doing, and just include it in the supplement, for example. But would people be able to really understand what you've done? Would they be able to use that information in a meaningful way? Probably not. So in this case, we can distinguish between two different types of issues. So there's the social issue where people simply say, well, I do not want to do this. Or maybe they forget. The other two issues that I point out are technical, right? We can address this. I mean, if you know how to work with computers, we can certainly find a way to capture everything we've done in the computer. And if you think about data visualization and using computers to communicate, we could maybe also find a way to actually communicate what we've done. But those are two separate problems. Both problems, though, are related to provenance. So what is provenance? Have you heard about the term provenance? Just out of curiosity. Has anyone heard about the term provenance? Okay, so what is provenance? So the term provenance has been used in a variety of ways to describe different types of origins and histories. So there are two pieces. There's an origin, where did it come from? And then the history, how did it get from where it came from to where we're now? So that has to be distinguished. And when we look at data provenance, we can distinguish different types of provenance information. So there's the data provenance. And that really just describes how data passed through, for example, an analysis pipeline. How did we get from some raw data to some derived results? On the other hand, when you're moving data through a pipeline, if it's a purely computational pipeline, well, everything is written down in some scripts or other terms of expressing the analysis workflow. But as soon as you start interacting with the data, for example, if you visualize the data, so like the subset in the data, we are moving into a different realm, and that is down here where we're talking about visualization provenance. How did we actually select this subset? There are many different ways how you can display a particular, say, tabular data set. You've probably learned about heat maps and scatter plots and profile plots. And if you want to capture how the user selected a subset of the data, you need to capture how they looked at it, right? So not only the fact that, well, they had this table and they picked the first 10 entries, for example, but in what way did they look at it? Because we want to understand maybe where things went wrong. So that's for visualization. Similar questions can be asked, or similar things could be captured about interactions with the system. Did you zoom in? Did you apply certain filters? And what colors did you display? Etc. And then on a more or even higher level, you can ask questions or capture information about insight and rationale. So in some cases, it's actually important to capture why we did certain things. So that's the rationale. And to incorporate that in what we write up about our analysis. Okay, so here we can distinguish, again, two groups. The data provenance is related to computational workflows primarily. And then we have the exploratory visualization tools that should be or would be responsible for capturing visualization provenance, interaction provenance, insight, as well as rationale provenance. Now, as you probably all know, there's a lot of back and forth between these things, right? So we run a computational pipeline, for example, going from some mass spectra to some peptides, or even protein quantifications, or we can sequence a genome, we can align the reads that we get to the genome, we can call variants in that, on those aligned reads, and then we could, for example, try to come up with some pathogenicity scores for these variants, etc. So usually there are many tools involved, and there are many steps in these pipelines, and at some point we'll look at the data and we start asking questions in the visualizations. And then often we go back to refine our workflows, for example. So that's this back and forth between computational workflows and exploratory visualization. Okay, so I want to give you an example of how we can capture and visualize provenance in computational workflows. I think this is something, are you all sort of doing computational analysis on a regular basis or occasionally? Just to give me a sense of, you are doing? Okay, all right, so in any case, I think this is, you could apply these kind of, or the issue that I'm trying to explain here doesn't only apply to data, it also applies to anything you do in your life or in a lab. When you do something experimentally, you have a protocol that you go through, many steps, and many parameters and things that you need to capture. So I want to show you this little video here. This is out of this Refinery platform. Which Refinery is it's a data analysis platform that we created to ultimately build visualization tools. Visualization tools that can directly interact with data analysis workflows. And so what it does really, it handles your data sets. You can load data sets into the system. You can run analyses on that data using a system called Galaxy, that some of you might have heard about, which is a popular bioinformatics workbench. And you can visualize the data. And what Refinery does when it comes to provenance and helping people interpret data is it captures all the steps that the analyst takes. So from loading the data in to capturing all the metadata associated with the files that you import into the system to capturing all the analyses that you execute on here. And this is an example of an analysis that sort of demonstrates what happens when you do when you run a genomic analysis pipeline where you have a QC step, you have a read mapping step, and actually this is for ChIP-seq. So we were looking, for example, at histone modifications in that data. We have several downstream analysis steps. And so this is an example of a provenance graph. So the relationships of the raw data that is over here, and it is very small, and that's the whole point of this. We have a lot of input files. Here's a branch where we did QC, and I know that you don't, and you wouldn't be able to see that if you were showing you this graph. Then we started doing some read mapping, peak calling, and then further analysis. So what is the problem with this? And I'm going to try to play this video. I don't know if this is going to work, if you'll be able to hear what I'm talking about. see.
Due to the size of this graph, there is no room for labels. Even with details on demand and zooming, it is hard to get an overview of what was done. However, due to the nature of bioinformatics analyses, this graph is highly structured and emphasizes by coloring each workflow type in a distinct color.
Now, as I point out, there were different workflows that were run, and as a first step, I'll just kind of show you where these different workflows are by simply coloring the nodes associated with these workflows. And the nodes in here is a tool, the inputs into that tool and the outputs of that tool. And usually it's a chain that's executed. So we had our raw data in blue. This is a QC workflow in orange. This is the read mapping in green. There's a workflow called Max2 in red. Corresponding workflow that does pretty much the same job called STP down here in purple. And then we have some pilot visualization down here that we've also run. So this is the first step. Just helps to get a slightly better sense of what's going on here. And where I'm getting at with this is a visualization problem in itself. So how do we, we've captured everything, but I'm trying to communicate to you what we've done, which is very important when it comes to reproducibility. And so this will demonstrate how we can take these big graphs, and because they have a particular structure generally in bioinformatics, based on how we do data analysis, we can collapse them into something much more understandable.
Information in this graph is hierarchically organized. We can show the graph at different levels of aggregation.
And now we can collapse them down.
Workflow instances.
These are workflows. Now we collapse workflows.
Analysis input groups. Analyses. And what we call layers that represent repetitive patterns of workflow executions.
Right. So what I was able to do, as you noticed or saw in the earlier view where we had the individual nodes for every workflow, there was a lot of repetition. Basically, the same workflow was run over and over, just on different inputs. And so we can exploit this and simply collapse this into a single node and say, well, everything was run through this workflow. But in this case, for example, the QC workflow, nothing came out of this. This is just a QC report that we want to look at and don't care about any further. But here, for example, we've collapsed all these read mapping workflows into a single node, and information that's relevant is here. You can't read it probably. There's a number on here that says how many edges are actually going in. That's a very large number. I think there are about 24 edges going in and 50-something edges coming out of this node. So now if we care about what really happened in here, we could drill into this, and I'll demonstrate that, and inspect that part of the workflow in more detail. And so this is this idea maybe came up in the last couple of days of overview first and then details on demand, and you can drill into your data rather than putting everything in front of the viewer.
We can learn more about the structure of the study by mapping other attributes to the node color. For example, we can map the analysis execution time and the darker node process them.
So what happened here is, just jump back to the beginning, that's unfortunate. Here, what I'm showing you here is, rather than mapping the workflow color onto these nodes, I just mapped the time when these workflows were executed onto the nodes. So this gives us a sense what was done recently. So these things, when you work on a data analysis project for a large data set, it can take several days, weeks, months potentially, and particularly when you send out your paper for review, you get it back, you got to do more analysis. So if you want to just get a quick overview, like what were the more recent analyses, you can see this. So the darker the node, the more recent it is. And the lighter the node, the older they are. So these were the things that were done at the very beginning, and this stuff down here was done more recently. So we can capture that kind of information as well and communicate it. Okay, so this is just how we collapse the graph. And now the next question is, how do we inspect this graph? And how do we drill into it? So we're interested in this node up there, and so we can look at another video.
Their node marked the change indicator. The orange asterisk indicates that not all of the analyses in this layer have the same parameterization.
So this is
Indeed, the two analyses in this layer have different numbers of analysis input groups, and their outputs are routed differently. We will drill down into this analysis to obtain more information about the workflow.
So we're going deeper into this graph, and we see the difference here is like none of the outputs here were fed into the downstream tool. But for this node,
to find out where the inputs for this workflow execution came from, we can modify the degree of interest function to automatically expand any highlighted path to the workflow level.
So this is something we did to come up with this approach, which, by the way, is called avocado. It's an acronym, I promise. It's just a very complicated one. When you inspect these graphs, and you try to interpret what have you done, a common question is to see, well, how did I get to this file? So this is the history. How did, through what tools did this file essentially pass from the raw data all the way to the derived result? And in order to implement this, we've essentially come up with a way to apply what we call a degree of interest function. So this is essentially the user telling us or telling the system what they're interested in. So because you're often interested in more than one thing or one kind of thing, there are different types of or interest functions that you could apply. For example, what I'm demonstrating here is I want to know sort of what happened along the path from the raw data to a derived file. So what we do is we expand only the nodes along the graph, but collapse everything else. And so the user can essentially define what they're interested in based on their actions or by applying these filtering functions. Here, we care about what is highlighted. We want to know more about it. So I click on this.
Once we select the inputs for the workflow that we are interested in, their path to the raw data is highlighted and automatically expanded.
Review what were the tools that were applied to look at this information.
We zoom in to investigate intermediate files and tools on the path from the raw data to our workflow of interest. We can also revert this functionality and trace all the output files generated from a raw data file of interest. Your node marked with a change indicator.
Sorry. So what I did at the very last step here, I did the reverse. First, I was interested in a derived file and I want to know where it came from, but you can also ask for a raw data file, what did it feed into? So you can revert this process. And there are many other ways of how we can express sort of interest. For example, we could use time as a factor. So then in that case, more recent things would be more expanded. And older things would be more collapsed. So we have different ways of collapsing this, as you've seen in the very beginning. We can really collapse it down into a single node, or we can expand everything out. And in between, there are different levels that we can look at. And all of these things can be controlled by the user. So I have to say, although this is implemented in the Refinery platform and we can visualize our provenance information here, this is also a research project. For example, one thing that we realized is users generally, when they try to express what they're interested in, they don't want to assign sort of fractions to these different features that we can incorporate in this interest function. So they generally want to say, well, I care about what is highlighted, and they just want to turn that on. And they don't want to say, well, I'm 50% interested in what is highlighted and 50% in what is old. This often it's not even clear what that would mean. So this is something that we are now going to simplify and focus a little bit more on what users really are doing with this in the end. Okay, so this is a way to look at data provenance. This is a very common problem. Many of these data analysis platforms capture information like that, but most of them are not able to really communicate it back. So there's no way for you generally to look at that, or if there is a way, it generally doesn't scale very well. And this is what we try to address with this avocado approach. And at the end of my presentation, there's actually a link to a paper that describes how we implemented this and how we came up with this approach in general. Okay, so let's look at visualization. That's why you're all here. So what is the role of visualization in data exploration? And for that, I want to just briefly summarize sort of the scientific discovery process for you. We can start with a hypothesis, collect some data in an experiment, and then somehow interpret the data and come up with some great insight, great discovery. And that generally, once we find something, that's already a starting point to ask more questions, okay? Now, it looks like a vicious cycle, but there is a way out of it, and that's the publication, of course. So once you have a great insight, you can write it up, and if you're lucky, you can get it published. And in that case, we use data visualization as a tool for presentation of information. And essentially what we're doing with visualization in this context is we use it for storytelling. So that is one thing we can do. Now, there is another major use for data visualization, and I'm sure you've learned about this in the last couple of days, data exploration. So data exploration is really the process where we're interested in generating hypotheses, where we're trying to come up with questions that we might want to answer, for example, using a statistical approach, which I guess people are doing next door right now. And in this case, we're using data visualization as a tool for pattern discovery. Right? We can generate hypotheses once we see something interesting, an interesting pattern, an outlier, or particular clusters, or weird gaps in the data. That always helps us to think about and come up with new things we want to follow up on. And that's where we use data visualization a lot. And that's also one of the reasons why data visualization has become so much more important in the last couple of years, because in the traditional scientific discovery process, we always started with a hypothesis. That was science. It's hypothesis-driven, and you come up with the hypothesis, and you design your experiment to collect some data to either reject your hypothesis or not. And you would deal with a fairly small data set. But nowadays, we can do things that are, in my opinion, way more exciting. We can just collect the data without necessarily having a very clear or well-defined hypothesis. So we can generate this big pile of data and start asking questions and try to come up with hypotheses about that data. And that's because data visualization is right here at the beginning of this process. It really has a much bigger role these days because it is such a powerful tool to come up with hypotheses, because we can look at data in an unbiased way. We don't have to compute anything ahead of time, generally, and we can just see the patterns. That's at least the idea. Okay, so what do we do with this now? I want to give you an example of how we can generate hypotheses using large data sets, heterogeneous data sets, in the biomedical domain. So the example that I want to talk about is the Cancer Genome Atlas. Who here is familiar with this project, has heard about it? Okay. So the Cancer Genome Atlas is a study that was launched in, I think, about 2006, 2007-ish, something like that. Funded by the NIH, originally by the National Human Genome Research Institute and the National Cancer Institute. And it was a big investment. The idea here was to collect genomic data from thousands of cancer patients to essentially come up with a molecular landscape of what is causing cancer, what kind of mutations are responsible for different types of cancers. And so the exciting part here is that in that project, they not only looked at genomes, they also looked at clinical data or collected clinical data and molecular data. And so I looked at that as wrong. So this is, although the main study is over, there's still a lot of stuff happening in terms of data analysis, follow-ups. It's a huge treasure trove for anyone working on genomic data, for anyone looking for a large data set to explore. And of course, it's an enormous resource for the cancer biology community. So the cohort here included more than 10,000 patients, or 10,000 tumor samples, I should say. Many of those were specimens that were from people who had already passed away. But for all of those, they were all characterized using a number of different assays. And so one of the questions that we can ask here with a cohort like this is, what kind of cancer subtypes are there? So for example, as you're familiar with lung cancer, for example, or breast cancer, almost any cancer type has multiple different subtypes. So before these large genomic studies, tumors were generally classified based on the tissue or body part where they arise, as well as some histological classifications. But nowadays, we can do a lot more in terms of understanding what is actually driving these tumors, where they're coming from, what kind of mutations are leading to them. So looking at this cohort, just to give you a sense of what kind of data we have in here, I want to start with telling you a little bit about the data that was collected. So for each patient, we have mRNA expression data. That's either sequencing-based, or primarily sequencing-based these days, so this is mRNA-seq data. We have microRNA expression data, also sequencing-based. Protein expression data, that is out of a reverse phase protein expression assay. So this is only for a subset of proteins, about 1,000 in general. Copy number variants derived from whole genome or whole exome sequencing data. Variant calls also generated out of either whole exome or whole genome sequencing data. DNA methylation data generated on an array. And then a lot of clinical information about these patients. For example, the lung cancer cohort, you can find out how much did these people smoke. You can find out all sorts of staging information, treatments that these patients were on, et cetera, et cetera. And the project team really did a great job in even following up on that data. So in some cases where there was missing data, they went back to the actual patient record to find out what these parameters were. So this is a very, very rich data set, and it's also a very large data set. So if you can imagine 10,000 at least whole exomes for everyone, plus all the other data, is very complex, and particularly because of the analyses that we want to run on that. And so what kind of things can we do with this if we are interested in finding cancer subtypes? And the subtype here would be defined as a group of patients within a cohort. So I should add that, by the way. There were 10,000, or there are more than 10,000 patients in the study overall, and they were spread out across about 20-something tumor cohorts. So for example, the breast cancer cohort has a bit more than 1,000 patients. Most of them have about 500, but there's some rarer tumors where there are only about 50 patients in these tumor groups. And so we're interested now looking at one of these, for example, a type of kidney cancer that we'll look at in more detail in a second. And we want to figure out whether there are certain groups of patients that have common mutations, and also then, based on that, for example, common mRNA expression patterns, or microarray expression patterns, etc. So there should be something that these patients have in common, and should also somehow affect, for example, outcomes. Do they live longer, or do they pass away faster than others? So when we have mRNA expression data, for example, we can cluster that data, and then we would end up with a stratification of the cohort based on gene expression data. So there might be 3 clusters, for example, C1, C2, C3, and C4, 4 clusters. Okay, so you could use any type of clustering on the gene expression data to get something like this. We can also look at copy number variants. For example, we can take any gene in the human genome and ask, what copy number status do these patients have for that particular gene? And we might see that in some patients, they have a deletion. It could either be a homozygous or a heterozygous deletion. There are actually 2 types of deletions we could look at. A lot of them will probably have normal copy number status, and others will maybe have an amplification. There could be a DCGA distinguished generally in their data sets between a low-level amplification and high level, so this is just the number of copies. So there, in total here, I'm showing you 3 different categories for copy number, but in fact, there are generally 5 available. We can take these same patients and ask, well, what is this mutation status for a given gene? So some patients will have a mutation in a given gene, and others, or majority, will be wild type or have no mutation in a given gene. We could do this in more detail and actually ask where in the genus a mutation. We would get a lot more categories out of this. But to simplify things, we're just going to look at is it mutated or not. And we would, or what we're interested in, is in essentially finding groups of patients where, or not groups of patients, we're interested in finding stratifications where we can find this kind of overlaps. Here this means everyone in cluster C2 also had an amplification in a given gene, as well as a particular mutation in another gene. So this is likely to be very rare that we see this, but we're looking at essentially these overlaps between these sets. So it's a set intersection problem, essentially, and we have many different ways in how we can slice these cohorts, and so we have many different stratifications of the cohort, and we're trying to explore that. And there are different ways in how we can do this. So we could just take an algorithmic approach, and for example, just look at all sorts of different ways of clustering the data, and then try to align across these columns. Well, we can also run some integrated clustering. There are methods to do that, where we can take many different data types and cluster them together to find patient clusters, for example. There are tools to do network-based stratification of a cohort that looks at the mutations in genes and then tries to generate a network and create some grouping based on that. We could also just, once we have these clusters, for example, run some correlation tests to see if there's any association between these groups. Okay. Now, the problem with this algorithmic stuff is, you can run the algorithms and you have all this data. You'd still probably want to look at it somehow to understand what this algorithm actually found for you. Which of these patients are overlapping? What does that mean, for example, when I find a cluster? What sort of information can I get in terms of patient survival? What can I get in terms of how are, for example, expression patterns different between this group and other groups? I might want to look at a pathway map to interpret that expression data. So there are many downsides to these algorithmic approaches. Others are, even though there are many approaches, there's not a single sort of super approach that would just do this analysis automatically for us. There's no way to automatically discover subtypes without human intervention, which means data visualization. So keep that in mind whenever we need to interact with the data. We're generally interested in visualizing it. Okay, so there are visualization approaches. That's the good news. For example, something called integrative heat maps. You can maybe have seen something like this. Here we're looking at a figure from a paper, and this is important. So the figure is in the paper. This means this is not an exploratory visualization tool. This is something after the fact, after the authors of the study, Robert Hack and his colleagues did this one. After they found, and after they were done with the analysis, they created this figure to include it in the paper. So what we're looking at is glioblastoma, which is a very aggressive brain cancer, 4 different subtypes, frontal, neural, classical, and mesenchymal. There are only about 100 and something patients in the cohort at that time when they wrote that paper. And what they listed up here, for example, are a couple of genes and their mutation status. So TP53 and its mutation status. So red means mutated, black means not mutated. IDH1 mutated versus not mutated. There's gene PDGFRA, gene expression levels for this gene, copy number status, and mutation status, and a couple of other genes. So what you can see, for example, there's this classical subtype over there, and the third one, the blue one. There are no TP53 mutations in any of these patients. So patient is a column in here, just to make that clear. There are no TP53, no IDH1 mutations. On the other hand, though, we see high-level amplification of the EGFR gene, which also seems to be reflected in the gene expression levels. So if you have more copies of a gene, you'd expect that you see more expression, unless there's something else that actually then basically downregulates its gene even more. So by looking at this, we can see some patterns. So the classical subtype in here, when you look at that, it looks quite different from some of the others, particularly in terms of the EGFR patterns, as well as TP53 and IDH1 mutations. When you look at the chronal subtype, you see almost half of all patients have a TP53 mutation. Okay, so this is great, but this is, as I said, after the fact. They already had the story more or less written out. They just wanted to demonstrate what's going on. There are tools such as genome browsers. For example, the IGV genome browser. I don't know if anyone here is familiar with this tool. You're looking at the same data set. The subtypes are now listed along the side here. Each row in here represents a patient. And so there's the classical subtype, the neural, proneural, and the mesenchymal subtype. And what we're looking at is data from 2 separate platforms. So these things here are sequencing-based, copy number data. And here we're sort of like only in a couple of spots that is array-based copy number data. Because this glioblastoma cohort was one of the first ones that they did. They didn't have sequencing data for all patients. They started out using arrays for some of these assays, or for some of these data types. So but what you see, again, very strikingly, the classical subtype, almost everyone has an amplification here, and here is the EGFR gene. So what this thing does not include, for example, are other data types. So gene expression data is not included here, as well as all the other information about mutation status of other genes. The downside of genome browsers is generally they'll show you one region in the genome. They can't show you many different regions. In order to do that, you go to something like a heat map approach. Okay, so that's what a genome browser does. Now, one of the downsides with genome browsers is, although this is an interactive tool that you can use for data exploration, so one downside is, as I already pointed out, generally with one region of the genome, not at many. And the other downside here is it's all mapped to the genome. So we can't easily bring in such information like patient survival for a particular subset. Okay, so what can we do about this? Let me recap the problems. So what we're trying to achieve here. So we need a tool that somehow can visualize these overlaps between patient sets. That's what I showed you in the schematic earlier. We want to essentially find these patient sets that somehow align with each other, where we have a lot of patients that somehow have something in common. The second problem is because even though we might see that they have something in common, the question then is, what do they have in common? How are they similar to each other, and how are they different from the rest? So we somehow need to bring in information about these patients' sets, about the patients in these patient sets in order to be able to interpret that information. And here is a tool that we built to do that. This tool is called StratomeX. That stands for Stratome Explorer, and Stratome is a word that we made up. It's supposed to be the set of all possible stratifications of a cohort. So as you can imagine, when we look at this data set, for example, right, we could cluster the patients on any of these data types. So that will already give us 5 different stratifications just by looking, say, at the microRNA, the mRNA, the protein expression levels, the methylation, and say copy number. And then, of course, we could apply any of the thousands of clustering algorithms that are out there, and they'll give us different results. So that already adds a lot more stratifications. We can also look at the copy number status per gene, right? Every gene in the genome and its copy number status in a cohort like this will end up with 25,000 different stratifications because every gene will stratify the cohort potentially in a different way. Same can be done for mutations. So we quickly end up with thousands of different ways of stratifying this cohort, and we are looking for patterns across all these different stratifications. So that's why we came up with this word Stratome and Explorer, just to indicate that we're trying to explore the set of all these different stratifications. So how does this work? It looks pretty, well, I don't know if it's pretty straightforward. In essence, what do we have here right now is mRNA-seq data clustered into 4 clusters. And here's an example for a gene BAP1 and its mutation status. So this is the kidney clear cell renal cell carcinoma data set from TCGA, about 400 patients. Within these blocks that represent a cluster, each row corresponds to a patient. The genes are columns. So typically when you look at a heat map, well, it depends, I guess, on what kind of data you're looking at, but often it's the other way around. Just to make it clear, a patient is here. And so here in this block, you can imagine there's some imaginary rows, each row representing a patient and their mutation status for this BAP1. So there's a small subset, about 10% of patients have a mutation in BAP1. We're actually summarizing this data type at the top, so you can see in this little block at the very top the distribution between mutated and not mutated, as well as the distribution of expression values in this column over here. So these are 2 stratifications of the same data set. And then we also have overlap. So when 2 of these sets overlap, we draw a line between them, as you can see, or a band. And these bands are, they correspond to the size of the intersection between, for example, this set, M3, and the mutated set up there. You'll also notice that there's a lot of missing data, essentially. There's no band here at the top. That simply means we have a lot of patients for which we have the gene expression data, but there's no corresponding mutation information. So not everyone who had this array done, or this gene expression analysis, or sorry, not for every patient in this case for which we had a gene expression data, we also have the mutation data. So that's a very common occurrence. As you work with these data sets, there's a lot of missing data. I also want to point out that just at this point already, you can see there's some difference across these clusters in terms of the expression values. Right? So the colors just mean it's a gene upregulated or downregulated. And you see, for example, in this cluster M2, there's some genes, we don't know which ones those are, that are definitely more highly expressed compared to this cluster M1 or M3 or M4. So this area here looks very different from the other clusters. So we can see there's some difference. We don't know what it means yet. And in order to figure out what these stratifications mean and what impact they have, we can incorporate other information. So for example, here, what we have in this column is patient survival data. So how long did they live after they were diagnosed? That's a Kaplan-Meier plot. Many of you will be familiar with this type of visualization. And what we've done here is, because that information by itself does not stratify the cohort. I mean, we could apply some sort of grouping on patient survival data, but we already have a grouping, and we can just look at our grouping. That is it BAP1 mutated, yes, no? And when we look at that, we see that patients who have a mutation in BAP1 have worse outcomes than those patients who do not have a mutation in BAP1. So we use an existing stratification to stratify other data types. And we can do something similar for gene expression data, for example. So because looking at these heat maps, you can stare at them, you can see there's a difference in the pattern, but you will not be able to figure out what that really means. So when we try to understand what that means, we often use tools such as pathway maps, where we're looking at a subset of the genes and their interactions and relationships. And we can do this in stratomaps by mapping data from a stratification on top of a pathway map. And so we get a different, we have the same pathway map 4 times. This is just a ribosome. And we can map the expression data for all the ribosomal genes out of this cluster on top of this map. So we average across all patients for every gene in this pathway. And what you see, a very striking difference. In here, compared to this cluster, so here red means upregulated relative to the mean of the cohort, and blue means downregulated. So relative to the cohort, to the average expression levels for the ribosomal genes in this cohort, this cluster M4 has the ribosomal genes highly upregulated, and this cluster M1 has them downregulated. And these two here, where it's sort of in the middle, means, well, there's some variation in there. And that's probably what you would expect. In general, what you expect to see is, just look at a bunch of random genes across a subset of patients, I would see, well, on average, it probably would be somewhere in the middle. It wouldn't be an extreme outlier. But the fact that we see very high expression here and very low expression here indicates there's something happening with the ribosome. Okay, so I already explained everything. Now, one thing that is very important when you look at these little maps, so you've probably heard about the small multiples concept that has come up in the last couple of days. I hope you all remember what it means. So this is great, small multiples, this is what we're using. What is not so great about it is it's small. So we don't really know which genes are highly regulated and which ones are not. So we have the ability to look at the data at different, essentially resolutions. So we have a very low resolution view on the data. But in the tool itself, and this is just a little video, I can expand open this view and I can get the map in detail. So now I can inspect the individual expression values as well, so we can hover over those, zoom in, zoom out, blah, blah, blah. So that's what's happening in here. And we can do the same thing for any of these blocks. For example, over there, we can not only expand out these individual cohort blocks, but we can also do the same thing on the summary blocks at the top. So here's the Kaplan-Meier plot for those patient clusters, and we can expand that out in a second, hopefully. Right now we get a detailed Kaplan-Meier plot where we see what these 2 lines mean, the percentage of patients, the days, how long they lived. Okay, so any questions about this so far? Okay, then I'll move on. We have a half an hour left. So problem number 3 is actually the hardest one. So what I just showed you were more or less 2 randomly selected stratification and just some data that we mapped onto these stratifications. But generally, the question is which stratifications are actually relevant, because we don't know what is driving these subtypes. So we need to have a way to find stratifications that overlap with each other. And this is how we can do this. So we proposed this approach called guided exploration. And the guided aspect here is that we use algorithms to guide the analyst through the data set. As I pointed out, this data set is very large. We have a database with hundreds of thousands of stratifications, many different clinical parameters which can be used like a stratification. So if I have a categorical variable across all my patients, I can stratify my patients based on that variable, for example, tumor staging information. Or they also capture information about race and other things. We could use age groups, etc., to stratify the cohort. And finally, we have thousands of pathways. So I don't know which pathway to look at, right? So we need to find ways to do this. And so essentially what we've designed is we implemented a query interface. So we use this visualization itself as an interface to enter a query against our database. So this is like typing a keyword into Google. This is the same question. But we want to use the visualization to define that query, because that visualization is a very expressive form of representing the data. And so if we can use that to define what we're asking for, such as, is there a mutation that overlaps with this cluster? We can run that kind of query directly out of the visualization. I'll show you how that works in a second. We can ask, is this mutation, or are there 2 mutations that are mutually exclusive? So that means, this is a very interesting situation in cancer. When you have a mutation that is essentially driving or causing a tumor type or a tumor, you don't need another mutation to do the same thing. So often what you find is that there are mutations that occur in one set of patients and another mutation that never overlaps with the same patient that is causing another subtype. So these are mutually exclusive mutations. we can ask questions, is there a copy number change, like an amplification or deletion that would affect survival? Or we can ask, is there a pathway that's somehow enriched in a cluster? So gene expression based enrichment. And then we want to rank all of our stratifications or other things we're asking for. We can rank pathways and stratifications. We want to essentially rank them, visualize the ranking, and then pick out those things that look good. And then we can just put them into the visualization. So how do we do these queries? There's a long list of algorithms. I don't want to go into detail that we can use. So we can look for overlap between sets. There's a measure called the Jacquard index to measure the similarity between clusterings. We can also ask, for example, are there two clusterings that look the same? So are two stratifications pretty much the same thing? We can ask that using something called the adjusted rand index. Survival can be done with the log rank score. And then we can use gene set enrichment approaches to retrieve pathways. Okay, that's not that relevant. What is maybe more interesting to you, because it's about data visualization, is the ranking. So to do this ranking, to visualize these rankings, which are generally based on multiple attributes. So a minimum would be the score, the retrieval measures that we use to essentially rank all our stratifications. But generally, you have other things in there that you might want to take into account. For example, if you're ranking genes, you can include categorical information. Is this a tumor suppressor gene? Is it an oncogene? You can include p-values, et cetera, all sorts of things to filter and explore these rankings. And here we developed a new technique called lineup to visualize multi-attribute rankings. That's a very generic method. So what I'm showing you here is a university ranking. As you know, these things are very popular, but they're generally just, you look at the US News and World Report and it says Harvard is number 1. Or Princeton is number 4. And then Princeton people say, why are we number 4? So in fact, these things are based on some metrics that they somehow magically turn into a number or into a rank. But you can get that information, actually. For example, things like student to faculty ratio plays a role. And these things are weighted differently. And we can visualize all this stuff in this visualization. And you can move the weights around and change things to see how your university would do, say if you would double the faculty to student ratio, et cetera. Often you have this information over time, so we have rankings across many different conditions or at time points. So this is 2013, 2012, all the way to 2008. You get different ways to aggregate that information. So here we see everything in this ranking, individual contributing factors, the same thing for the next year. And then we have can aggregate this into sort of just a bar chart or into a heat map kind of visualization. And so this tool is very popular. Microsoft has a platform called Power BI, stands for Business Intelligence. They integrated that. So it's open source. Anyone can go to the website down there, play around with it. You can easily load tabular data into that tool. It's nowadays web-based, and you can explore your rankings based on that. Okay, so we developed this approach in the context of Stratomax to rank and visualize our query results. So this was a nice side project. I want to show you how this querying works. So to run a query in here, we developed something called a query wizard that allows you to define what the question is that you want to answer. And so here our question was, is there any tumor suppressor gene that happens to be deleted in this cluster? So we can do this. I'm going to show you a little video. So first of all, we need to select the cluster we want to query. We start by adding a new stratification, and the question is, how do we do it? Based on overlap, okay? So we select the target, we want to overlap with this thing, and boom, we're already done. So we have a ranking down here that shows us similarity of the copy number call on a particular gene, how it overlaps with this cluster over here. And so this is not a good hit. None of these clusters really overlaps, or none of these copy number changes really overlaps very well with this cluster M3, but we still get a ranking. And as you can see, we get a preview. We can just go through that list. It's very convenient. We don't have to worry about saving things and not being able to undo anything. So I'll show you it again. So you want to add a stratification. Yep.
Sorry, what is it ranking by?
So it's ranking based on the overlap. So this is using the Jacquard index. Basically looks at, we have two sets. You can look at the intersection and the union. So if the size of the intersection is the same size as the union, you get a score of 1, which is the best. It's perfect overlap. And if it's smaller than 1, which means your union is larger than intersection. So that's what we're doing. We're comparing this cluster to any of the copy number categories, basically. We're looking across all genes and all possible states. So amplification, normal, or deletions. So we're comparing about 150,000 different sets to this set here. So this is the target, and then this is computing this, and here's the score. Similarity score is very low, so 1 would be perfect. And I'll show you some more striking examples in a second. So yeah, we can sort of go through this, and you see there's this red X and the red green check mark. So whenever we find something that we're interested in, we can confirm this, and the stratification will get added to the view. Okay, so now we can use this approach to explore one of these TCGA tumor types. And this is the kidney renal cell clear cell carcinoma. We have about 400 patients in this data set or in this cohort. And our question was, well, there was already a paper published on this tumor cohort by the TCGA project that we were part of. And they had described some subtypes in that data. And our question was simply, well, can we find more subtypes? Can we find groups of patients that are somehow different, behaving differently, that maybe, and of course, the rationale is to go back and find, specialized or more appropriate treatments for these patients, or to characterize at least these subtypes in more detail. So what we did, we essentially looked at methylation data, microRNA-seq data, mRNA-seq data, and the reverse phase protein array data to see if any of these clusters that we get out of that data, if there's any group of patients that has worse survival than the rest, okay? When we did that, we found that there was one clustering. So we had, for all of these 4 data types, we had clusterings between 2 and 10 clusters. That is just data that TCGA was generating by default for every of these data types. And what we found when we ran our query, we just asked, is there any cluster with worse outcomes in any of these clusterings? We found one, and that's the RPPA, reverse phase protein array, clustering with 8 clusters. We found there's one cluster in there, number 8, that has much worse outcomes. So this was based on ranking using the log-rank test. This was the first hit. So you see, here's our score. It's very high. Also, when you compare it to the next level, there's a big difference. So this is the stratifications, the scores, and then some other information here. So this is what we found. We're like, wow, this is really striking. Looking at all the other cluster groups in here, these patients have much worse outcomes. And so you can do this with an algorithm, and it'll tell you that. But now the question is, why is it, and what do we find? So I'll tell you, I'll take a little tour here. So the first thing we wanted to do, as we do generally, is we try to figure out, are there any genes or pathways that are somehow affected? Now, the very first step we had to do is because we realized these things here are all pretty similar, so we didn't care too much about those. But there's a feature in StrataMax that allows you to merge these things. So we merged these 7 other clusters into one, and then we have basically cluster number 8 and the rest. This is what you see over here, and you see the striking difference in survival times for these patients. So that's the first step. The next step was related to genes. Because we have only about 1,000 genes in here, and the number of patients are the same, but the number of genes in these columns here is much lower for the reverse phase protein array than it's, say, for the gene expression data. So in order to run analysis such as gene set enrichment analysis, we need more genes. We can't do this with 1,000 genes. So we wanted to have more genes, and what we tried to use the RNA-seq data as a proxy. So here's the protein data, here's the gene expression data. And here you see there's not really much, it's not a clear difference in terms of the clusters here. Neither do we see this in the RNA-seq data. The only thing that this step allows us to do is to run queries on pathways. So we did this. We took this cluster number 8 in our gene expression data and asked, are there any pathways that are differentially enriched or differentially expressed in this cluster number 8? And here's the ranking. Down here, you probably can't read this. Among the top 10 hits here, we found 3 pathways that are related to DNA repair. We thought it was pretty interesting. So homologous recombination, base excision repair, and then mismatch repair were all somehow differentially expressed in this cluster number 8 compared to the rest. So here are these 3 pathways from familiar with the tag maps, and you know that we can drill in and see detail. And in fact, there is definitely some differential expression happening. So this cluster is highly upregulated. There's several genes in this cluster that are upregulated, while there are others that are downregulated. No, these are all upregulated. This blue here means something else, unfortunately, in this map. And here are a couple of other genes that are also upregulated in this cluster up there compared to the rest. Okay, and so yeah, we can drill in. We could have studied these genes in more detail, but we postponed that because we had another question. Now, knowing that DNA repair pathways are highly expressed or differentially activated, we're wondering, is there a difference in the mutation rates in these two groups of genomes? So we can get the mutation rates from TCGA. So by looking at the exome data or genome data, you can look at the frequency of mutations, and they can also distinguish between silent mutations that would not have any impact on the protein level and the non-silent mutations. And so we took the same idea as before. We have our stratification, we have our other variable. In this case, it's the mutation rate. We just stratify that variable into two groups. And then instead of the Kaplan-Meier plot, we just plot a box plot. And as you can see here at the top, there is a difference. So the top cluster is the one that we're interested in, cluster number 8, this box plot here. And there is, in fact, this difference that you can see here, this shift. There's actually a significant difference in terms of mutation rates between these two groups. And we got really excited about this until we looked at this. So we ran another query. We simply asked, is there anything that has a big overlap with this across all our different stratifications? And one of the stratifications in our data set is the staging information. So there are different tumor stages, stage 1, which is early and not so bad, up to 4, which is very late generally. And the cells are very undifferentiated, and these patients generally are not doing very well. And so turns out, after ranking this stuff, we found that this stratification and stage 3 and 4 have a very high overlap with this cluster. It's hard to see this here, but there are basically only stage 3 and stage 4 patients in this cluster number 8. That's why they have so much, that's why their outcomes are so much worse. Okay, so at this point we were really disappointed because that simply meant, well, this is not a great finding, although what is demonstrated to us and then luckily also to the reviewers and the editors of Nature Methods, that this approach actually can work to find interesting hypotheses. And the nice thing is it not only generates hypotheses, you can also quickly follow up on it. So this whole analysis took only half an afternoon to get to that point. So before that, you would have to load this data into R and run all these algorithms and generate your own plots. And here we can do most of that pretty much on the fly. The only thing that we were not able to do directly in the software was the significance test for the difference in mutation rates. So now what? It's great. So we found this thing and we wanted to include this in our paper. And that's where things got really hairy. As you remember, up here, right, we managed to do this. We were trying to basically go from our discovery to selling that discovery to, well, not selling it necessarily, but to telling the world about what we found. So how would we do this? And it turned out, in order to do this, for this publication, we generated a very short article and about 50 or 60 pages of supplement that had screenshot after screenshot after screenshot, the description of what we did, how we got, because our job was to capture our exploratory process. We wanted to tell the readers how we got from the starting point all the way to the end. So we not only had screenshots and text, we also had a video, and you saw part of that video earlier. So it's about 10 minutes of video to tell part of the story. And it was incredibly tedious because in order to make it reproducible, we had to write everything down, exactly how we did these things, in which order, what parameters to use, et cetera, et cetera. And that is a problem. So we wanted to basically go from data-driven discovery to data-driven communication. How can we use our data and the visualization tools that we use to do the exploration to actually communicate this stuff? So how do we traditionally do this? And you just saw it. Traditionally, we have data exploration, we make these discoveries, we find these interesting things, and we want to write them up. And then we create a figure or video and we want to present that somehow. So the presentation could be a paper, it could be a presentation like this, it could be a piece of paper that you hand your boss or your colleagues when you just have a meeting to get together. Authoring generally happens in tools like Illustrator, Photoshop, or PowerPoint or something. And then exploration happens in R and specialized tools, etc. Whatever you use to do your data analysis. And in this model here, information flow are the black arrows, and the gray arrows indicate how you can essentially go through these stages. You always start here with the exploration, then you move to authoring, and you move to presentation. But there's no arrow back, as you can see here. Well, and what's the problem with this, right? So number one, this is the figure we show in the paper. Generally, besides the figures, we also say a lot of things in the paper. Often there's no corresponding figure because journalists don't like that when you put too many figures in, and you can only make them so small. So there's generally more that we're talking about than we can show in figures. And what's even more important, this is really what we did. Generally, we don't go straight from our hypothesis to our figure. Usually there are a lot of dead ends, a lot of exploration and trying all sorts of things. And that's really an issue, because this is really what was important. That is what we did, and that's what led us to our finding, right? There might be, knowing about which different routes you took can be very useful for me to reproduce what you've done or to understand how you got there. There could be many other branches in here. If you don't tell the world about it, they might ask NIH for funding, and NIH will give them the money and run into the same dead end that you run into. So knowing about all of this is very useful. What do we need to do to actually capture all of this and do this, right? So first of all, we need to track provenance, as you know already. We need to annotate our observations, because just looking at what I've done or what you've done will probably not completely get the message across why you've done it. So annotating things is important. Of course, we need to interpret all of this and then tell the story. So we call this capture, label, and understand, which creates a nice acronym, CLUE. And so what is this CLUE model like? So in CLUE, this is really a model. This is not, well, this is implemented, but this is just how to think about it. So in CLUE, there's one data structure that captures everything. All your observations go into that data structure. All your authoring is done on that data structure, and all your presentation is driven by the data structure. So rather than having X different things, we do this on one object, basically. And the nice thing is, when I have everything in one place, I can actually go from my presentation back to my exploration, where I can go from the presentation to authoring. So I could go in and refine something. So how did we do this? So we implemented this model in the tool that I just talked about, in StratoMax. So now this is a web-based version of StratoMax. It's not feature equivalent, but very similar. So there are 3 different modes. We call them modes because they're not really stages at this point, because we can go back and forth between these different modes. So in the exploration mode, we have our StratoMax tool over here. Here's sort of the data browser. Here's the visualization. And over here, we have a provenance graph that captures or includes everything we've done in here. When we switch from one visualization to another, this creates a new state in the provenance graph. When we add more data, it goes in here. When we filter the data, it goes in here. So that's all happening in there. And you can see this is not linear, right? Because the exploration is never linear. There are many side branches in here of things that we tried, and then you can always sort of like an undo, jump back to your previous state, continue from there. And when you do this, you will create a new branch in this graph. So once we're done with our exploration, temporarily or permanently, we can go to the authoring mode. In the authoring mode, we have the same stuff. In addition to that, we have a story editor. So what is a story here? Essentially, a story is how you got from your input data to your conclusions or to your findings. And that corresponds to a branch or to a path through this graph. Now, of course, there are often multiple stories that you want to tell, but that means you can extract multiple stories and add in multiple stories. And really, a story is literally nothing but a selection of nodes in here, generally consecutive nodes down a couple of branches that you convert into a story. You can time these individual steps if you want that, because everything in here can later be replayed like a video when we go to the presentation mode, where we essentially have one of the stories that were created, individual steps listed out, and you could either push the play button or step through it manually. So the timing is not that important. One thing I want to point out is that in the exploration, and so in the authoring mode, this is here, I think. Oops, no. Sorry. Hey. Here's the authoring mode. In the authoring mode, we also have annotation tools. So we can, this is where we add information about what we've done. If you want to explain something, you can put an arrow on there, or text, or a box, etc. So all of that goes in there. And yeah, then you have the presentation mode. And just to show you that again, in the presentation mode, although we're in presentation, at any point in time, you can click on authoring or exploration up there, and then go back to exploration. What it does, wherever you are, right? So when you're in presentation, each step in here corresponds to one node in the provenance graph. And if you go from here to the exploration mode, you'll just go into that node in the graph, and you can either go backwards or forwards and do whatever you want to do. So this is really, really useful if you are sitting in an office with your colleagues and you show them what you've done, and at some point they say, hey, why did you not look at this thing? And if you're doing this with this tool, you can look at this thing. You just jump back and explore from there. And so this is sort of the traditional workflow on the right, the thing that we painfully went through once and said, we're never going to do this again. So what we're doing now, we're essentially creating all our visualization tools that with this clue model built in. So all our visualization tools, ideally, not all of them have it at the moment, will support this approach. And what we call these things that are coming out at the end, so this thing we call it a vistory. And this vistory is a visualization plus a story that you're telling, plus its history. So that's the vistory. And we've implemented this in these two demos at the moment. There's several other tools that are partially under development. Some of them are already under publication or in review right now. The second tool you might be familiar with is called Gapminder, which is a public health data exploration tool that is very popular. And you can use these things to do collaborative analysis if you just want to share with your colleague what you've been doing. During peer review, that's very helpful too if you want to show the reviewers what you've been doing. You can include it in a presentation, so I didn't do this today, but in theory you can create a HTML-based, web-based presentation where each of the slides could correspond to one of these vistories, and I could just jump in right from there and continue exploring. And you could publish that with a paper as well. So in the interest of time, I could play this video, which is essentially a little demo of how we use this for Stratomax. I think it's only about a couple of minutes, so 3 minutes. So we were interested in looking at mRNA, microRNA expression profiles, copy number, mutations. Same story basically that I already walked you through. But what we're doing right now, we're just replaying one of these vistories. So this is now unfortunately captured in a video, which sort of defeats the purpose. But for presentation purposes today, I had to turn this into video. But literally, you could go to the website and replay this whole thing and explore on your own. So the first step is we add stratification, and here it is. Here's the annotation. There's just a view, and then we continue through here. And after a while, so I'm going to jump forward a little bit. So I think you get the sense. We can put these annotations in to explain what I'm doing. I guess I could have just shown you the video earlier. That would have saved me some time and energy here. So that's the whole idea, really. You want to communicate about your exploration, about your data, using visualization. This is the way to do it. So we're going through here all the same things that I did earlier, looking at different types of variables, survival, categorical variables, gene expression data. We can rearrange the columns, etc. But now at some point we'll switch over to the authoring mode or exploration mode in this case, and we're right there, and we could continue whatever we want to do. We can ask whatever questions we want to ask at that point. Right, and so this will create a new branch in the provenance graph. Okay, so this is how we go from data-driven discovery to data-driven communication, and how we actually go back from data-driven communication back to data-driven exploration. So as I said, we have a couple of demos, prototypes. When you go to vistories.org, there is both the Stratomax demo, which I noticed this morning has a slight bug at the moment. So it works in general, but some of the heat maps look a little funky. There's also the Gapminder tool that you can play around with, which is a simple scatter plot where you can map different variables onto your data. Sorry, I don't know why I repeated this. I guess it's important. Here are two papers describing some of the work that I discussed today. So the first one, Avocado, our provenance graph aggregation and exploration tool, and here is the Clue and Vistories paper that you can check out, and I'm going to share the slides with you somehow, I guess via PDF. I want to thank my collaborators in Austria and the University of Utah, who have worked on there's actually about a half dozen projects that I talked about. The other papers that I probably should have mentioned are the lineup paper, the Stratomax 2 papers. So this is all the team of people who were involved in this as sort of the core researchers. Also need to thank various funding agencies and institutions for their financial support of my work. So in particular NIH, of course. And finally, also, in case there is anyone who's looking for a job in this space, I'm hiring postdocs. Also should add, Harvard has a new biomedical informatics master's program, if this is of interest to anyone. That's an 18-month program that includes a lot of data science, among them visualization, data sciences, and visualization, of course, is one of them. And with that, I'm happy to take more questions, or any questions, in fact. And I can also show you some more demos if you're interested. Thank you very much. Is anyone interested in seeing a demo? That's an easy question. Okay, so I'm going to stop my presentation now, and I'm just going to stop the recording. Just you.